All right, so before many of you came on, Sandra asked the appropriate question, when would you get feedback on your first summaries? I've done about 2/3 of them, so I'll be giving those back to you in the next few days. So you should have it in plenty of time to write your second summary this weekend before next Monday when we have our second paper summary. Um, if you've looked at the syllabus, you can see we're kind of behind. We're about a half a lecture behind. So we definitely will not be doing sampling and enumeration and molecular diversity methods today. We may not even make it through the sampling and enumeration after we finish up extremophiles. But we do have a little wiggle room. We'll have a review discussion on March 1st before the midterm on that. That's the Monday before March 3rd is a Wednesday. And if we need to finish up a little bit of lecture, we can do it that day. And then you guys will also have the opportunity to bring questions to for me, and I'll kind of go over what to expect on the tests at that point. So let's go ahead and get started. So we left off talking about the extreme environments, and we left off right before hypersaline habitats. As you know, we talked about these, but this will give you a little bit of context of some of what we read about and discussed in the paper. So in the background here, you can see actually a picture of a saltern. So this is what that Spanish saltern site would have looked like. You can see the red color and the brines that are there. Many of these are coastal salterns. So what they do is they pump seawater into a series of ponds, evaporation ponds. And as it gets more and more salty, it gets redder and redder. And then ultimately you end up with these, white salt ponds where the halite or the sodium chloride precipitates out. So if you're using seawater, we call them thalassohaline. But there are some cases where there are like salt ponds or lakes that are derived from geological salt. So, there are caves that have salt and sort of old salt. And these often have a very different chemistry to them. So excuse me. So for example, the, why is it doing that? The Dead Sea and the Great Salt Lake up in Utah are more magnesium rich than, as opposed to sodium rich. There's also brine ponds in Antarctica where the freezing that they get seasonally, freeze-thaw cycles, will create salt ponds because the freezing mimics evaporation. Anything that's hypersaline would be greater than 35 parts per thousand, which is what we see in seawater, about 33 to 35. Remember, we use that parts per thousand. That'd be about 3.5%. Okay. And there's often high primary productivity of both eukaryotic algae as well as heterotrophs that have photopigments, and we'll talk about that. So who are these halophilic microbes that live in these really salty environments? This is a picture from the Guerrero Negro saltern that I did much of my postdoctoral research in. This is a gypsum precipitating pond. And the halophiles that we see there are a combination of all three domains. So there's a green alga, which actually is sort of hard. It's kind of a misnomer. It's in the green algae lineage, but it looks red because it produces carotenoid pigments, and it's called Denaliella. So they contribute to the red color that you saw in the previous slide. There are many Archaea, including some of the ones that we saw in the paper, like Haloarcula, Halorubrum, and then the square bacteria, square archaea, Haloarchaeans shown here, which is Haloquadratum. Bacteria also are found in many of these hypersaline environments. They're typically not the record holders in terms of high salinity. Those are usually the Archaea, but you can go, upwards of 2 molar salts, which is very salty. And many of these are groups of organisms that are found in the ocean, but that can tolerate or thrive under saltier environments as well. like heterotrophic groups, um, like Halomonas, Selenivibrio, and Cyanobacteria. And then there's another group of bacteria called Selenobacter, which is, they seem to have laterally acquired a number of features from halophilic Archaea. And so they often are found in the very highest salinities alongside the Haloarchaea. So how do these organisms survive in this environment? They don't have the membrane issues that the thermophiles we talked about have, but they do need to worry about osmotic stress. And they do have things going across their membranes and cell walls. They will often use sodium if it's found in abundance, as it is in many of these environments, to help stabilize their cell walls. There's two main mechanisms for balancing or dealing with the fact that the water that you're sitting in is super salty, right? Water or, uh, it can also be like salt crystals and things like that where organisms are found. So some organisms will pump, and this is primarily the Archaea, will pump potassium inside. And I think that was mentioned, we briefly discussed that the other day, and it was mentioned in the paper. So they take potassium up from the environment, pump it inside the cell, and balance the salts inside and out in that way. That many bacteria, on the other hand, don't pump salts in. What they do is they synthesize sugars like glycine betaine or trehalose. And it turns out that even though they're in a salty environment, if you have high sugar content, that also creates a osmotic pressure. And so you can balance out the osmotic pressure that way. This doesn't work quite as well in the really extreme salinities where the active pumping is required. Another key aspect found primarily in the Archaea is the production of something called Bacteriorhodopsin. Now, I know that name suggests that it came from a bacterium, but remember back in the day, we didn't know that Archaea existed. So back then they thought, in fact, one of the earliest studied halophiles was Halobacterium, which turns out to now be an archaeal genus. So Bacteriorhodopsin was named for these organisms that under low oxygen conditions, which are actually quite common in salt ponds, where because of the salinity gradient, oxygen diffusion can be even lower than normal. And so in their membranes, they produce this carotenoid-like retinol molecule called rhodopsin. Now, you guys have heard that term rhodopsin before, because where do we have rhodopsin molecules?
In our eyes.
Exactly. So they don't have eyes, of course, but what they're doing is using these rhodopsin molecules to absorb light in a similar way to what we do with our rods and cones, right? Instead, what they're doing with it is generating ATP. So they have a proton motive force that's generated. So there's a conformational change that occurs after they get excited when the rhodopsin molecule is hit by a photon of light, and this generates ATP. This is really important if you're going to be constantly pumping potassium inside the cell to balance your osmotic pressure, for example. You need lots of energy for that. And under anaerobic conditions, when respiration rates are going to drop, this can help keep them active that way. So that's a very rapid overview of halophiles. Now we'll move on to acidic environments, but are there any questions about the halophilic adaptations or organisms? All right, do not do this at home. This is a picture I always like to show because it's just crazy. This is a quite old picture from that guy, Tom Brock. Remember I told you about Taq polymerase and how Dr. Brock from Wisconsin gave Thermus aquaticus away or whatever. Well, there were many researchers that would go back in the 50s, 60s, 70s to do research. You can see these guys are in suits with hats. It's quite old, of course, but it's crazy what they're doing because many hot springs that are acidic are essentially sulfuric acid baths. right? Or pools of sulfuric acid, and that's what you see here. So the fact that this guy is like holding his jacket as he's dipping his thing in to sample this hot spring is just, if you were ever caught doing that today, you'd be immediately kicked out of the park, of course. And it's made even worse because the ground around acidic hot springs is really rotten, and it's really easy for it to give way or for you to punch through. So it's just crazy. Another very common habitat for acidic conditions are volcanic soils. And then perhaps the most important in California um, is up is acid mine drainage. So in many places like California where deep rock mining occurs, right, as we know happened during the gold rush, when they drill holes into the mountains to extract gold or other minerals, they let air and oxygen in, and this creates a set of chemistry that leads to acidification. Okay, so those three environments are the main ones that we'll focus on for acidophiles. Who are the microbes that live in these environments? Well, as you may remember, if you took intro micro, for those of you that are micro majors, in general, fungi tend to be a little more acidophilic than other prokaryotes, but they're not majorly acidophilic in most cases. So the real acidophiles are either algae or prokaryotes. So there are two different kinds of these algae, Cyanidium, which is a red alga, but ironically it looks green, and Zygogonium, which is a green alga, but it looks purple. There's also Archaea, and these guys, again, are sort of the record holders. So you can see a pH of almost 0, this Picrophilus. They're also a thermophile. So these are the kinds of organisms that live in these environments. Perhaps the best studied and most famous acidophile is called Sulfolobus. Sulfolobus is isolated from a hot spring, just like what I showed you with the crazy guys dipping their scooper in. These are aerobic sulfur oxidizing thermophiles. So they live in acid hot springs all around the world. There are also bacteria, though, that can also experience lots of low pH environments. And many of the acid mine drainage organisms are in these groups, Thiobacillus and Leptospirillum. So you're going to actually read the Tyson paper, which will describe one of these acid mine drainage situations a little bit. So I'm asking you to read, I asked you to read that for today as kind of like a supplemental. If you haven't had a chance to read that, please do take a look at it. We're not doing a summary on that paper, but it's just good background. The opposite of acidic environments, of course, is alkalophilic. Does anybody know where this picture was taken?
That's Mono Lake, right?
It is, yeah. Thanks, Jenny. Have you been there, Jenny?
I have. It's by like by Mammoth, right?
Yeah, so if you go up to Mammoth, it's kind of like a little bit past and out east of the Sierras there. So Mono Lake is one of the world's best examples, but there are a number of soda lakes or bicarbonate lakes, and that's what's creating these mounds or tufas that they are called that come up out of the water, and they're basically precipitated bicarbonates. Um, there are also quite a few of these lakes in East Africa where the Earth is actually splitting apart in the rift areas of East Africa, where eventually, millions of years from now, that part of Africa will separate and there'll be an ocean between it. But in that area, because of all the tectonic activity, it turns out there's quite a few of these bicarbonate lakes too. But certainly Mono Lake is our most locally accessible one. Now, remember that neutrono that was found in the halophile paper that we just read? So neutrono as a prefix usually indicates the organism is an alkalophile. And so remember, some of those sites were actually alkaline. And so many of these are hypersaline, and high pH. And so you end up with these organisms like these Archaea, Natronobacterium, again named before we knew what Archaea were, or Natronococcus. But there are cyanobacteria and other bacteria like gram-positive groups like Bacillus or Clostridium gram-positives, spore formers that are commonly found. So there's actually quite a diversity, and there's been research done like in Mono Lake on the diversity of microbes living there. So how do these organisms survive under low or high pH physiology? So just taking a step back, most organisms, us included, usually have a fairly narrow pH range. Microbes are usually a little broader than us, and so it might be 2 to 3 pHs, which, remember, it's orders of magnitude. So that's hundreds of fold difference or thousands fold difference in proton concentrations. However, most organisms maintain an internal pH near neutral or 7, right? So if they're living in an environment that's pH 2 or 3, they have to figure out how to adapt to make sure that their insides don't turn acidic, or in the case of alkalophiles, they don't go up to 8 or 9. So again, because you're dealing with things wanting to go across a membrane or a cell wall, just like we saw with the salts, membrane adaptations are important. And so they use protons. So we saw that halophiles use sodium to stabilize their cell walls. Here, the protons are actually used to stabilize the membranes in acidophiles. So if you're being bathed in protons, it actually makes it really hard to generate a proton motive force. So what they do is they've adapted to use the protons to stabilize their membranes, and then they substitute sodium they generate essentially a sodium motive force instead of a proton motive force. And this is commonly found in both acidophiles and alkalophiles, just because the chemistry is so extreme, shall we say, in both extreme acidic and alkaline environments. All right, so let's have our third poll question of this lecture, but the first of today. So we've talked about ions in a number of different contexts here. So changes in the use of ions for important structural or metabolic processes were found in which of the following? Was it in hydrothermal, hypersaline, acidic, both A and B, or both B and C? Hopefully you can all get this. I think this might be the easiest one of the semester so far. All right, a couple more seconds. Go ahead and log your answer. All right. Okay, so the vast majority of you got it right, putting E, both B and C, because we said that the cell walls were stabilized using sodium from the environment in hypersaline or halophilic environments. And then the acidophiles are going to use protons to stabilize their membranes and generate proton motive force with sodium. Okay, does everybody see? I think most of you got that right. All right, the final thing I want to share from this, and this is a topic that's near and dear to my heart. Unfortunately, over the years, I used to have a single lecture dedicated to this, but because of the scheduling, I've had to shorten this over the years. So we just get a few minutes to talk about microbial mats. Microbial mats are, they've been around a long time, as we've already talked about this semester, but nowadays we tend to find them in extreme environments. So either in the intertidal zone and coastal environments or in hypersaline lakes and ponds, they're very common, where the salinity either fluctuates or is consistently high, because basically to have a microbial mat or biofilm, these things are macroscopic. You can see them with the naked eye. It's a huge amount of biomass. And to have that situation where they don't get eaten by eukaryotic organisms, grazers mainly, or outcompeted by eukaryotic organisms, they need to be in these so-called extreme environments where they can adapt and do well, but many of the eukaryotes are more limited. We also see them in thermal environments, and I've already showed you pictures of hot springs with cyanobacterial mats that I've studied before. You can see microbial mats, not photosynthetic mats, but you can see sulfur chemolithoautotrophic mats near those hydrothermal vents. So not all microbial mats are photosynthetic, but many of them are. In cold temperature environments like polar ponds in Antarctica, we can also see these mats because the cold temperatures restrict grazers and other eukaryotes. So this is a picture that just shows some of the lamination. So this is a core sample taken down into the mat. So at the surface, you have cyanobacteria, but you can see all of these different layers. Some of these are mineral layers, like these white things, but many of these are layers that are created by the organisms living together. So there are cyanobacteria, but there are also other phototrophs and oxygenic phototrophs that don't produce oxygen that will live near or below the cyanobacteria in these microbial mat environments. And you can see that here. This is a picture I took from the Guerrero Negro Saltern, where I studied these microbial mats, and you can very clearly see the laminations. Some of this is mineral, but most of this is biological. You can tell by the pigmentation. Now it's interesting. This is when you leave this out overnight and it oxidizes. But when I first pull these mats out, look at the difference. This is all hydrogen sulfide that is produced in these mats down deep. And if you leave it out in the air, then the sulfide oxidizes, and then you can see all these layers. So there's very complicated chemistry, and it's often sulfur-based, particularly in those hypersaline systems. So these are complex assemblages, very diverse, lots of organisms living together. Where there's cyanobacteria, many of these are filamentous cyanobacteria that have lots of interesting morphologies, like these corkscrew-shaped ones and long chains. There are other phototrophs like the anoxygenic organisms I was mentioning that are filamentous as well. But there's whole, suites of dozens and dozens of species that live associated with them that you can't really tell what it is under the microscope, right? So typically we use molecular analyses to gain an understanding of this. And this is just a schematic put together by some collaborators of mine that shows kind of the diversity of organisms and where they're found. This is using that hypersaline mat as an example. So you see like cyanobacteria up here, other phototrophs down below, the sulfate reducers I showed down here, but there's also chemolithotrophic sulfur bacteria that live at the boundary between oxygen and sulfur produced by the sulfate reducers. And you can see if you look over here, there's a gradient that's established of oxygen and sulfur. And in the daytime, the oxygen production goes up, so the oxygen penetrates further into the mat. And then at night, the oxygen turns off, and so the sulfide starts to come up, and sometimes it even comes all the way up to the surface of the mat. So some of my own research in the Guerrero Negro mat tracked these chemolithotrophic sulfur bacteria, among others, and found them to migrate up and down in a day-night cycle because they were tracking their chemistry where they can do their metabolisms. So it's very diverse, lots of groups, and very dynamic systems on a diel or day-night cycle. All right, so that is a very brief introduction to microbial mats. Any questions about that?
Yeah, I have a question.
Sure.
So, just like we kind of talked about right now, oxygen and sulfide stuff from the environment is affecting the, that mat being sustained. In terms of all those microbes living together, do they have like a kind of like mutualistic relationship? Like, do they each support each other, or is it just like environmental factors that keep it up?
No, it's definitely lots of symbioses and mutualisms, right? So the cyanobacteria and the sulfur oxidizers are carbon fixers, so they're bringing carbon in, but they're feeding many of these other organisms, right? Because they leak carbon out of their cells or they die and they lyse and liberate it. So you have lots of heterotrophs, both aerobic organisms up in the surface as well as anaerobic organisms like fermenters and that are going to use that carbon. But the nitrogen cycle is complex, the sulfur cycle is complex. And so there's lots of interactions that are really fascinating to investigate. And there's quite a lot of research that goes into trying to tease that apart.
Thank you.
Yeah. Yep, good question. So let me close that. I'll go ahead and move on. So we're now switching gears to talk about methodologies. Okay. So what we're going to do for the rest of the time before our midterm is we're going to talk about methods that are used to study microbes in all these amazing habitats that we've gone over. And if you're going to study organisms, you got to get samples first. And so the first lecture today is on sampling, and then we're going to go into some of the methods of how you count organisms. And so we already saw a little bit of that in the Oxenreiter paper that we read, right? So they took samples from those 3 different sites. And we'll talk today about how maybe they could have improved. I think we already alluded to that. And then they also counted cells, and we'll touch on some of those methods they discussed in the paper as well. So before you do any sampling or any research for that matter, you need to make a plan, right? You can't just go out half-cocked and just like pick up a sample and bring it back and decide, hey, I'm just going to study this, right? You really need to create a plan so that you can systematically investigate whatever question you might have. And so a quality assurance plan is part of that. It includes the sampling, but as well as like, how do I know that the samples are going to be properly maintained to ensure sterility, et cetera, et cetera. So the first thing we'll talk about in this quality assurance plan is the sampling. And we need to think about the numbers, the types of samples we would take, location, where are we going, as well as how often. And that came up with our Oxenreiter paper as well. A number of you asked the question, which was a good one. How come they did samples at 2 different times a year? And why did they only sample 1 time in each of those sites, right? So they certainly, that was a potential limitation of that study. And so if you were interested in questions related to time, you might want to change that. And another aspect of what they didn't really do in that study was they didn't really replicate their samples, right? They were just grabbing single samples in each of these locations. And so they may not have been as truly representative of the diversity as they were kind of letting on in their study. So to sample, we need to have specific methods that are used. And then, of course, we're going to then analyze using another suite of methods, okay? And then we need to store our samples. So you need to store them potentially in transit if you're going out into the environment to sample or in the field, and bring them back to the lab where you're going to study them. And so you have to preserve them. And so do you refrigerate them, put them on ice, maybe dry ice? Do you add fixative? Like, do you add formaldehyde, right? It will kill your sample, but it will preserve those cells for like microscopic purposes. And then duration, like how long can you store your samples before you use them, right? So it's interesting because quite a few of these logistics are very similar to some of the refrigeration and freezing logistics we're dealing with distributing the COVID vaccines, right? So how quickly you get the vials and how long you can store them at what temperatures. This is very analogous to that. All right, so we're going to have a breakout session. And what I'd like you to do is just, it's kind of a thought experiment. So as a group, I'd like you to come up with just a few ideas. If I ask you to go out to a lake and sample it for its algal diversity, so like micro eukaryotic algae, how would you sample that lake? And we'll come back and share, and we'll see if we have some ideas of how you might want to do that. So I'm going to stop sharing. Go ahead and set up the breakout rooms. I'll do 5 today. Great. Yeah. All right. Let's go ahead. So we'll take about 5 minutes. Hey, Marwa, were you able to join it?
Yeah, sorry, the little thing didn't come up for me. I was waiting for it.
It should have. Let me just try putting you in a different room.
Okay.
Maybe.
Okay, now I came up. Thank you.
Okay. All right, and then let me. All right, and then Valerie, I can get you put into Caden's room.
Thank you. She's in 1, I think.
Yeah. I don't know why it's not. There you are. Okay.
Thank you.
Yep. All right, so welcome back. Hopefully you guys had a chance to have some meaningful dialogue. Does somebody from group 1 want to share one of the thoughts you guys talked about in terms of what you would do? If you remember if you were in group 1. Or let's just rephrase it. Somebody want to share? Anybody?
Yeah, we talked about taking various samples at different depths, taking a soil core sample, sampling around different regions of the lake, shaded, sunny, different times of day, night and day.
Okay. So you're really like hitting a lot of the different aspects. So spatial variability, depth, seasonal, or day
Right.
of time, or okay.
We were group 4, by the way.
Okay. Other groups, did you guys have a different plan? Johan?
Yeah, so group 5, we discussed about storing the samples in formalin for algae to preserve the cells. And we discussed taking samples from the topmost layer or different layers of the cell lake to find the composition of that layer. And we talked about use of sterilized equipments.
Okay.
Take samples.
And so why did you guys talk about doing the top? So that was a little different than what Victor talked about in terms of sampling down deep as well. What was your rationale?
So some algae might be oxygenic, like they use a lot of oxygen for metabolism and use of sunlight as well. Whereas there might be some algae which might be found in the deep lake where light may not reach much more as compared to the topmost.
Yeah, I mean, I would expect most of the eukaryotic algae to be up near the surface, like you were saying. By the way, let's clarify. Oxygenic means they produce oxygen, not use it, right? So it's always confusing, right? Respiration consumes oxygen. Photosynthesis produces oxygen. So they're not using the oxygen up there. They're actually making it, but they are using the sunlight, right? And the reason they produce oxygen, if you remember, is they're splitting water to get the electrons, right? And that liberates oxygen during that process. So yeah, okay,
question.
Cool. Yes.
So will oxygenic organisms be there on the top, you mean?
So some of the anoxygenic might be near the top, but many times they are found lower in the water column because some of them are obligate anaerobes. And so they actually need to live down where maybe there's some of that sulfide being produced in the bottom of a lake, for example.
Got it.
So, okay. All right. Anybody else want to, share what your group talked about? Or does that kind of cover most of what each of the groups talked about? Anybody talk about something different?
I guess one would be like the type. So that's kind of something that we kind of like try to elaborate on, which was we see kind of like biofilms. We would try to like take from that, but also just water samples as well.
Right. So many of them will be planktonic, right? Just floating around in the water, but you could have biofilms in shallower areas that might be worth sampling. So great. Cool. Well, there's no like perfect answer for this. So I'm glad you guys were able to kind of reason through, because this is what you would do. And this is actually similar to what I'm going to ask you on the midterm for this portion of the course. I'm going to present you with scenarios and then ask you, how would you sample for this specific organism? What are the kinds of approaches or methods you would use? And so this is a good start for that, to get you thinking in that way. So replication is one of the things we mentioned. And really there's multiple ways of thinking about replication. If you think about it at all, of course, which I hope you do. So you can replicate either randomly, right? Using like a random number generator type thing, or you can replicate haphazardly. So like if you went down to the beach and I told you to collect seawater at the edge of the water, and you went and you collected some water, and then you walked a little bit further and you collected some more water in another tube, and then you walked a little further and collected a little more. That would that be random, or would that be haphazard, do you think?
Haphazard?
Exactly. And it's not to say that it's a bad way of doing it. In fact, many cases I've done that myself. Random requires you to like think about it in advance and figure out, map out a sampling strategy somehow, right? So oftentimes what we need to do is combine, particularly if you want random sampling, you're sampling with some sort of grid or a transect. So a transect is a line. You basically lie a line down on the ground or up a tree or whatever you're studying. So for example, you can take a big tape measure and run it. So if I had said, go sample at the beach from the water line up to the dunes, right? You could take a line and just run it all the way down. And then maybe you could say, I'm going to sample every 10 meters for the 100 meters between me and the ocean. This is particularly useful if you're sampling along gradients, which like a beach would be a gradient, right? A gradient of sort of seawater influences, gradient of slope, oftentimes, things like that. You can be more systematic and you can lay out an entire grid over certain systems, right? And so maybe this is what you would have done. It kind of sounds a little bit like what group 1 was talking about in terms of like sampling in different parts of the lake, maybe. And so you could set that up as well. And then we need to think about it. Does this grid have 2 dimensions? Does it have 3 dimensions like depth, as was also mentioned for the lake, which is good? Or if you're taking a core sample from a soil, that would have depth as well. And then the 4th dimension, of course, is time, right? And so do we go back and sample every day, every month, every year, or do we just take one sample and we ignore time as a factor, right? So these are the kinds of things you need to think about. There's no one way to do sampling. It really depends on what your questions are, what the logistics are, how much access you have to your site, and these various factors come in. But in general, we think of the more sampling, the better, okay? So when we're talking about sampling, if we're out in the ocean or in a lake, you can sample water with like a bottle, okay? And that was essentially what was described in the Oxenreider paper for a couple of their sites, right? If you're sampling the mud at the bottom of a lake, as shown here by my colleague, this is a picture actually out at the Salton Sea where we did some research years ago. And you put the core down into the mud and you can see there's a little water on top, but the rest of it is the muddy sediment from below. This would be what you refer to as a simple core. I'll show you a picture of a piston core on the next slide or in a couple slides. And then you can also sample sediments using something called a dredge. Okay. So before we get to some of those specific sampling contraptions, let's have another poll question. And this is basically just kind of getting you to think about this. So of these 4 habitats, which of them do you think would be the easiest? And that's kind of a loaded term, I realize, but which would be the easiest to get a representative sample? Like if I said, go out and take one sample in these 4 habitats, which would be most likely to be truly representative of that sample or that location? Okay. All right, I relaunched the poll. So what do you think? All right, go ahead and log your answer if you haven't already. Okay. So you're kind of all over the map here, but it looks like most of you went for the dirt sample. So either soils or a lake sediments. So somebody who put A, can you explain your rationale? Why do you think that's easiest to get a representative sample? Like if you could only take one soil sample and analyze it, this is what you're choosing.
I'm not sure if this is correct, but I thought because of the picture you showed, with the simple core example, I feel like that would have been like you can use it with soil. And so I thought that that would be kind of easiest. I was between soil and lake sediment, but I went with soil.
Okay. So it would be kind of easy to get a sample.
Yeah, and also because I think we said that soil has like different components. So I think it would kind of have like a lot of diversity. So I think it would be a good sample.
Okay. But that raises the question of, okay, so if I only get to take one core, and there's a lot of sort of diversity, and we talked about sort of patchiness and variation in soils and sediments, that could create some challenges too, I think, in terms of that. There's no perfect answer for this, by the way, so I just am always interested to see what you guys are thinking. Somebody who put seawater, what were you thinking? I know it was only 3 of you, but it's okay. If it's any consolation, I would put seawater actually as my answer.
I don't think it varies much across. I mean, I don't know. I just don't think it varies that much when
And
thinking
why
of
doesn't it?
seawater.
And why do you think of it not really varying?
The conditions like the salinity, maybe
Okay.
the salinity is and pH and all that stuff. Maybe it's just consistent all over.
Okay. And maybe it's less impacted by like rain or weather or things like that. Is that kind of where you were?
Yeah.
Coming from, Carlos. Okay, so yeah, and there's mixing that goes on. So, obviously coastal seawater is very different than open ocean seawater. So, you do have to kind of, maybe I should modify this but in some ways, a seawater sample is going to be more homogeneous than a soil or lake sediments. But of course, if you wanted to sample the Sargasso Sea, it's not necessarily easy to get out there and take a sample. So that's sort of tricky. Only 2 of you put the deep subsurface, and I'm guessing that was just because of the difficulty of accessing that. Is that what most people were thinking? But if you chose deep subsurface, why did you choose that?
When I was answering it, I just thought it was like this. It was highly specific because I thought like soil, you could take a top uh, sample or deeper, and I don't know,
Right.
deep subsurface seemed pretty specific. So I thought it would be less.
So if
less
you could
to have to deal with.
Like if you could get down there, then it would be pretty easy to get a sample that probably represented all of the subsurface samples then. Okay.
Yeah, that's what I was thinking when I answered.
Yeah, it's interesting to sort of think about it. I'm not going to have a question like this on the test. I just like to get people thinking about the issues related to sampling and what might come into play. All right, so here are some of those contraptions. Oops. So this is another push corer that's being used in this little stream here. Very commonly used in wetland kind of environments like you can see there. For deeper samples, a piston corer is often needed, and particularly if you don't have leverage to push down. So if you're 50 feet down in the water or a kilometer down, you've got to find a way to be able to get that core into the soil or the sediment at the bottom. And so a piston corer actually, as the name implies, has the ability to drive the core through a mechanism, right? Like an explosive mechanism that pushes it down. And typically this is triggered remotely from the boat that you're lowering this down for. All right, looks like we've got some answers in the chat. Yeah, Jenny, there wasn't really a right answer for that last clicker question or poll question. So I chose seawater, but I think it's fine just what you wanted to have. All right. And then I mentioned those dredges. These are clamshell dredges that are commonly used in lakes. You pry open the bottom there, like he's pulling that up. You drop it down, and then again, you send a remote little trigger, which causes these restraining straps to break free, and it closes and grabs some mud from the bottom. Now you can also drill or auger in, as shown in this picture, right? And this can be a great way for you to get samples like from a soil or sediments. But we have issues to consider whenever we're taking that. So one is contamination. You don't want to have, as you're augering in, for example, if you take organisms and dirt from up here and then shove it all the way down and into your sample as you're augering in, then when you look at it, if you think that dirt is from down here, but it really came from the top, then that's an issue. So you have to use these tools properly and know how to avoid contaminating samples. That's also true in the deep subsurface. We worry a lot about contamination of samples because they're very simple ecosystems down there, and it's potentially really easy for you to bring microbes there. Okay. Compaction is also like if you're digging down, if you use like that simple corer that I showed you, the picture of the guy from the lake, what you do is you shove that into the ground. Well, what that does is it squishes the sediment ever so slightly at least, or it can sometimes be pretty profound. And that leads to compaction. So then let's say you want to look at layers in a soil and you want to go down, 10 millimeters or 10 centimeters into that soil and look at what's there. Well, if you had compaction, then maybe what was 10 centimeters is now at 8 centimeters. And so it can throw you off. And a lot of these issues of contamination and compaction can be driven by how much water and moisture content there is in these samples. So we need to think about that. Nonetheless, we can pretty reasonably collect sediments and soils using those different corers or augers. For sampling for water samples, aquatic sampling, the sampling itself tends to be easier. You've got this more homogeneous nature of the water, although we did talk about particles being sort of almost a hidden but important component, particularly in the oceans, right, with marine snow and things like that. So it's visually more homogeneous, but at a microscopic level, it may not always be. It tends to be a little bit easier to get samples there, as I was saying. That's why I would have chosen seawater for my samples. But of course, that's not necessarily true if you need to get on a ship and sail, miles and miles out into the ocean. You can get your samples remotely, and I'll show you some ways that we do that as well. But there are some challenges, right? You may have to filter your water to concentrate it, okay? Particularly out in the open ocean or in very pure like mountain lakes where the water quality is really good, you have low biomass, you may need to filter hundreds to thousands of liters of water to get a sample that has enough biomass to analyze it, right? Another thing to think about is, viruses are ubiquitous, but if you're interested in studying viruses, they're very small, as you know, and you may need huge volumes to be able to analyze that. And so they also stick to things or adsorb. So you may have to consider the issues of them sticking, and then you can actually use that to your advantage, though, and get them to stick to a membrane and then elute them or liberate them again later and reconcentrate them in smaller volumes. So oftentimes you can filter, or what you can do is use an adsorption apparatus to concentrate them. So for water samples in general, we tend to use things like this, which is a Niskin bottle. What you can see is there's these rubber stoppers that go on either end, and this is just a cylinder. You lower it down off of either by hand, or they have a little boom here, lower it down, and then what you're going to do is send a trigger down that'll hit this white thing, causing the release of these straps, and then the little stoppers will fill the bottle. So what you can do is you can lower down your Niskin bottle. Let's say you want to take a sample at 10 meters in Long Beach Harbor, right? And this is actually what I do with my marine microbiology class that I teach in the fall. We go out, we lower it down, and then you trigger that and close it up. And then you've got this packet of water that you collected at a certain depth. Now, this works really well in lakes or near shore oceanic settings. But if you're going to sample out on like an oceanographic vessel in the deep ocean, then you often are going to use what's called a rosette. And here you can see there can be upwards of like a dozen bottles on there, which allows you to trigger the sampling of these bottles at different depths. And let's say you want replication, so you could trigger 3 bottles at the surface and 3 bottles at 100 meters and 3 bottles at 400 meters, or whatever it is you're planning to do. The other thing is that these are often attached to what are called CTDs, or conductivity, temperature, and depth loggers. And so a CTD will give you chemical information, and it's actually, it usually samples more than that. You can get oxygen, depth, salinity, chlorophyll, all sorts of metrics that allow you to kind of profile the water as you go down. And so you might be able to find like the 1% light level, and then use the CTD, find that, and then trigger the rosette to close some bottles there, for example. More simply, if you're just interested in, let's say, some of those algae, like we were saying, I was asking you to talk about in the breakout, maybe you just do a plankton tow at the surface. And what this net does is it just funnels algae into a little bottle, and then you can look at it. And you can estimate how much makes it in there by how long you tow it through the volume and estimate that. Now, filtration is very commonly used with aquatic samples, but one of the problem, and you may have encountered this if you've ever filtered something that has particles before, if you use sort of standard dead end filtration, which is what you've done in like a chem lab or a biology lab, where you just draw a vacuum and pull things through, what happens is as the particles that are in there fall to the filter, they will clog the filter. And what that does is it slows the flow. Has anybody been filtering something and suddenly the filter like stopped, the water stopped going through? In any lab you've ever been a part of or anything? It's actually pretty common if you're going to be studying microbes like bacteria or algae on a filter. So tangential or cross flow filtration was invented a number of decades ago. And what this does is, instead of having the feed stream go straight down, it actually puts the particles in a stream going laterally or tangentially. And so then what happens is, over time, water is able to permeate and leave the system, concentrating the particles, but they're not clogging all of the holes up like they do if you put the pressure this way. All right, so that is a very quick overview of sampling. Any questions about either soil or sediment sampling or the aquatic sampling? I know I'm going kind of fast today, so I want to make sure you guys have a chance to ask questions if you have them. Okay, so the next topic is enumeration. And so our question is, how many critters, how many bugs are there in my sample, right? So we can do this with puricultures, and those of you that are even in general biology lab, I think we do this, where you read absorbance or turbidity in a spectrophotometer, and you can figure out, oh, I can count the amount of E. coli in a tube that way. If I know what the absorbance is, I plate it out, count some of the colonies on a plate. I know this is a streak plate, not a counting plate, but this was the picture they had. But the idea is you can count them fairly readily, either in liquid, which is here, or on a solid medium. by counting colony forming units. Okay. And so many of you have done that repeatedly in your careers. The problem as microbial ecologists, though, is if you want to study the environment, we don't have pure cultures mostly, right? And so if you go out to like a lake and you collect that water, you can't just put the lake water in a speck and count the microbes that are there, right? Because there's debris, there's cells, there's different kinds of cells, and all of them will affect turbidity. You can try and put them on a count, but it can be hard because we don't know how many of them are viable or alive and would grow, for example, on medium or something like that. So that said, we can still try to take some approaches and we can use viable counts and compare them to direct counts. So viable counts typically are looking at living and often culturable organisms. Direct counts, they don't have to be living or culturable. So let's first talk about these viable count methods. And before we get to sort of how the methods work, I want to bring up the name for what we've already talked about repeatedly this semester, and that's called the Great Plate Count Anomaly. So this is a term that was actually coined by a former mentor of mine, Jim Staley at the University of Washington. He and some of his students named this. So this is the phenomenon that if you grow things in culture, most things are not culturable, right? So if you want to count the organisms in an environment, and you did that by plate counts, where you take a sample, put it like do a spread plate on top of some sort of agar plate, right? Let's say it's seawater agar that you use, and you grab a seawater sample from the ocean. Fewer than 1% of the microbes in that sample are going to be culturable and grow on that. And so that was what the anomaly was. It was the distinction between what's there and what can be counted on a plate. Okay, does that make sense? And so many of these cells, remember that term? Does anybody remember what the VBNC stood for? We introduced that term a number of lectures ago.
Viable but not culturable.
Exactly. So these may be living cells, but they're not culturable, at least not using the culture media that we use. You can try and tweak things like in the Oxenreiter paper, where they tried to modify the salt concentrations to mimic the salinity, the chemistry of the sample. But as we saw in that paper, they were able to culture in some of the habitats, they could culture some of the groups, but not all of them, right? And then in some of the habitats, they really didn't culture much of anything that was related to what they got from the environment. So we know this anomaly is important. How important is going to vary from one site to another. That said, we can use plate counts or other cultivation-based approaches like the most probable number method, which I'll show you in a couple minutes. It's particularly effective to do this if you want to count a specific organism that you know is countable and culturable, right? And so oftentimes we're going to do this with selective growth medium. You just have to know that whatever is growing there doesn't represent like the true abundance of whatever that organism is in the environment necessarily, or at least it's omitting the abundance of many of the other things that aren't growing. So it's more an indicator of the potential for growth or the presence of organisms. And sometimes that's a really important thing to know. So for example, at the beaches, right? Every day, the sanitation districts go out to the ocean and collect water samples, and they plate them. What are they plating them for? You guys know? Are they plating them for obscure marine microbiology samples, or why does a sanitation district collect water at the beaches every day?
maybe to inform the people if there are any red tides or anything that might, or any other pathogenic organisms that might be found near the
Yeah.
shore today.
Yeah, so it's not usually red tides. What kind of pathogens do you think we particularly worry about? You're absolutely right, Johan, that it's pathogens. Does anybody? Why shouldn't you go?
Giardia? Sorry.
Oh, go ahead. Yeah. So Giardia is more commonly found in like mountain streams. It's less common
Okay.
in the ocean. I think we're overthinking this a bit. So why don't you go in the ocean after it rains? At least I hope you don't. What ends up in the ocean after it rains here in Southern California?
Runoff from the farms and the rivers and everything like that. So maybe existing bacteria in the water.
Yeah. So, and sewage is really the important thing, right? So human waste ends up being a huge part of what ends up in the ocean. So the sanitation districts are sampling for coliforms and other fecal indicator bacteria. So every day they go to the beach and they sample for things like E. coli and Enterococci, and they post and close. I guess you've probably all been to a beach where they said, oh, the beach is closed because the water's dirty, don't go swimming, right? This is the reason that happens is because the sanitation districts call the lifeguards and say, close the beach. Our samples from this morning came up contaminated. This is pretty common after the rain. Um, Although a lot of people, especially surfers, ignore that rule. But so these methods can be really useful in some contexts, but they're not giving total counts. For that, you really need direct counts to do that. And I'll talk about direct counts in a couple of minutes if I have time. Plate counts can be done a number of ways. You can do pour plates where you're mixing the sample in with molten agar medium, or you can take a solidified agar plate and just spread it around the surface. And you may be seeing those little hockey stick like things that you can spread water. If you do the pour plating, temperature selection is a part of it because you have to do it before the agar hardens. And so that may impose some temperature, some mild temperature selection. But if it's not thermophiles that you're doing, mild temperature problems could occur. Now this works well. You can put plates into an anaerobic jar to culture anaerobes, but some anaerobes are really fastidious, like sulfate reducers, and they don't grow well on a plate in those anaerobic jars. So many times you have to do anaerobic tubes that you gas with like nitrogen gas to get rid of all the oxygen for organisms like that. And so roll tubes are basically you put an agar film on the inside of a glass tube and then you gas it and it has a stopper. So we mentioned the targeted enrichments earlier, and this is an example. So the EMB medium, there's a number of different media that are used for fecal coliforms that I was describing earlier. But you can also target any number of other organisms as well. Most probable number method is a serial dilution method of a liquid sample. And so this is very commonly used to try to get a rough sense of the total culturable, let's say, bacteria in a sample, not necessarily pathogens. This could be anything. Let's say you have a seawater sample and you want to know how many culturable microbes there are. So what you do is you take your sample, in this case, it could be seawater, and you are going to do a dilution series. This is a 10-fold dilution series, but you can also do higher dilutions. And what we're doing is we're looking to see if there's growth in the first tube and the second tube and the third tube. And as you get more and more dilute, if you see growth, that means there were more cells in the original sample, right? Because if I had 10 cells in here and I dilute them 10-fold, I would expect to get growth here. But if I do 10-fold again, I might get growth, and then I wouldn't expect growth here. But if I had 1,000 cells in this original milliliter of sample, then I would expect to see growth here and here and here and maybe even here. So the serial dilution allows you to estimate how many cells per mil were in our original sample by counting how many positive tubes you have as you go out. And so you're using statistical inference or probabilities. And if you want to be accurate with this, you really cannot have a single series. So typically, a most probable number estimation is done with 3 or 5 sets of tubes. So you would have 1 series, 2 series, 3 series, and then you would count the dilutions and where you see growth in the 3 1 to 10s and the 3 1 to 100s and the 3 1 to 1000s and so on and so forth. Does that make sense? Are there any questions about that? So the more cells you had in your original sample, the further out in the dilution you would expect to see positive growth. All right, so let's go ahead and have another poll question as we finish up today. All right, so which of the following would be an explanation? And I didn't necessarily talk about all of these, but think about which might play a role. An explanation for that Great Plate count anomaly. Remember the less than 1% cultivability issue. Is it the inability of us to mimic environmental conditions in culture? Is it a lack of species-species communication? The time period for growth may exceed the times typically allowed for testing in the lab, or D, all of the above? Which of these do you think might contribute to the Great Plate count anomaly? All right, go ahead and add your vote. All right. All right, most of you put D, all of the above, which is absolutely correct. And I hadn't really talked about this. I had mainly alluded to A, right? So we're not very good at using a medium that mimics exactly the conditions out there. Even if you put the salts in like Oxenreiter did, it's still in a tube. It's still a different environment that it was in that Saltern, for example. But it turns out that there's likely lots of reasons, right? So organisms communicate with each other in ways that we don't really even know in many cases. And if you separate those organisms, then neither of them may survive, or maybe one does, but the other doesn't. The other thing is that when we're doing culturing, oftentimes we're going to do cultures for a set period of time. Do you guys remember how long the Oxenreiter paper did the culturing for? Anybody remember?
Was it 2 weeks?
Yeah, it was on the order of weeks, right? And that makes sense, right? You let them grow, and halophiles often do grow a little bit slower. But there might have been organisms that would have grown if they left them for a month or 3 months or a year that would have been different. And that's the important thing to note, is that not all organisms are going to follow like a 24-hour news cycle kind of, I need my answers now sort of approach. And so that can be another thing that causes that limitation. All right, so we will stop there, and